So today my talk will be the replication study of plasma biomarkers relating to Alzheimer's pathology. First, I'd like to talk about the Alzheimer's prevalence. Currently, there are 75 million people getting dementia, and this number is expected to increase to 135 million by 2050. Among all the dementia, Alzheimer's disease is the most common dementia, as you can see from this pie chart. The blue color shows you the Alzheimer's disease, accounting to 60 to 70% of dementia. The rest of dementia are vascular dementia, Lewy body dementia, frontotemporal dementia, Parkinson's disease, and so on. There are two main pathologies in Alzheimer's disease. One is A-beta, one is tau pathology. Both two proteins started to accumulate in brain, leading to the disease. So in terms of the cascades of the pathology in Alzheimer's disease, this is a classical figure which summarizes all the pathologies in AD. So in this figure, the x-axis is time development. Different color shows you the different pathology. For example, the purple color shows you the CSF A-beta, which means the A-beta level in cerebrospinal fluid. The red color shows you the A-beta measurement by PET scan. The blue color shows you the tau pathology in cerebrospinal fluid. The orange color shows you the MRI measurement, which means the neurodegeneration, whether the brain has a shrink or not. The green color shows you the clinical diagnosis using cognitive measurement, memory measurement, and so on. So as you can see that the clinical diagnosis range a lot from normal, cognitive normal individuals to MCI is a stage between normal and dementia patients. From this figure, you can see that Alzheimer's pathology, including A-beta, tau, neurodegeneration, happens 10 or even 20 years before clinical symptoms appear, which means that when a person complains about his or her memory, it means that A-beta or tau or neurodegeneration happens much earlier compared to this clinical symptoms appear. So for AD diagnosis, currently from pathological point of view, as I have briefly introduced previously, we could have PET scan to measure brain A-beta and a tau burden. However, the PET scan is very expensive. We could also do lumbar puncture to get a cerebrospinal fluid to measure A-beta and a tau level. However, this method is very invasive. It can cause pain to the patients. Thirdly, we could use MRI to measure whether there's a brain neurodegeneration. As you can see, the left hand is a healthy brain and the right hand is a disease, AD disease brain. So MRI is good, but it cannot be specific to Alzheimer's disease because it also happens in other neurodegenerative disease. From a clinical point of view, the doctors often use in clinical assessments, for example, MMSE to diagnose the patients. However, this diagnosis is not precise and also it's often too late. In comparison to this method, the blood test is a much more practical and cost-effective screening tool because it has lots of advantages. For example, it's non-invasive, it can be used for the early diagnosis, it's easy to perform, and can be done repeatedly if necessary. Because of this advantage of the blood biomarkers, our group have identified lots of protein biomarkers associated with the various AD pathology, including A-beta PET scan, A-beta and tau levels in CSF, MRI scan, and cognitive decline. So we have identified proteins which can reflect this pathology. When I first joined the team, I first wrote the review, summarized all the studies we have done during the last decade, and published in Journal of Alzheimer's Disease. As you can see from the title, it's called A Decade of Blood Biomarkers for Alzheimer's Disease Research. In addition, I discussed how to improve the study designs and how to cope with the challenge of replication. Actually, the failure of replication is one of the main challenges of biomarker study, because as you probably know that lots of people reported lots of biomarkers in the literature. However, few of the biomarkers can be replicated by other researchers. So that's the main challenge. In this review, I've summarized all the studies we've done, and I also, most importantly, discussed how to cope with the main challenge of the replication. So if you are interested in, you could check this review for more details. So now I give you, do you have any questions? If you have any questions, please stop me, or you can ask the questions after my talk. Okay, I'll continue. So now I'll show you 4 different studies that we have done for identify and validate biomarkers relating to Alzheimer's disease. The first study I want to talk about is called EMIF AD study, which is a case control study. The second one is called GS study, which is a community-based study. The third study I want to talk about is a meta-analysis, how we gathered all the data that we have and run a meta-analysis to test the robustness of the biomarkers for diagnosing Alzheimer's disease. And the last study I talk about is called DKK1 study, which combines in vitro and human samples together. So now let's look at the first one, the EMIF AD study. The EMIF is short for European Medical Information Framework. So this framework included 14 European countries, combining 58 partners from both academia and industry, as you can see from here. The whole platform has 15.6 million worth of resources. We reused the existing data and created 3 projects in one, including EMIF platform, EMIF metabolic, and EMIF AD. So now I focus on EMIF AD study. This slide shows you the timeline of EMIF AD study. As you can see, this study was initiated in June 2014. Initially, we invited 16 cohorts to take part in this study. After the QC, we signed 11 contracts. After then, we transferred all the samples to one center to do the experiments. We harmonized all the clinical data. We finished all the wet lab analysis in June 2018. And the rest of the time, we are doing the data analysis. So in detail, this study included 1,221 subjects, including nearly 500 healthy controls, over 500 MCI patients. MCI, as I mentioned before, is a stage between healthy control and Alzheimer's disease patients, and 202 AD patients. The most important thing about this study is that each subject had a beta measurement, either by PET scan or CSF A-beta level. In addition, we did a multi-omics for these subjects. For example, 97% of the subjects had a plasma sample, so we did both plasma proteomics and metabolomics. So for proteomics, we used 2 main approaches. First, we used the SOMA scan to test 4,001 proteins for each single individual. Second, we used the immunoassay to validate 34 previously identified proteins in this cohort. For metabolomics, we use mass spectrometry. 63% of them had cerebrospinal fluid samples, we also did proteomics. First, we measured core AD markers, including A-beta, tau, p-tau, NFL, YKL40. So these are the Alzheimer's-related biomarkers. Second, we use mass spectrometry to identify new biomarkers in CSF. In addition, 80% of the samples had genomic data and 17% of them had MRI data. So as you can see, this is a large effort from European collaborators. And at Oxford, we focus on plasma proteomics. So now I'll show you the results from proteomics analysis. I'll first show you the results obtained from SomaScan, and then I'll show you the results from the immunoassay. For the SomaScan data, we first analyzed the association between proteins and their beta. We divided the whole cohort into two groups, showing figure A and B. We used a linear regression and created for covariates such as age, ApoE, full genotype, gender, and so on. So as you can see, in group A, we found that 301 proteins which are significantly associated with their beta. In group 2, we found 536 proteins are significantly associated with their beta. We then did cake pathway enrichment for these dysregulated proteins, and we found that 10 pathways are significantly enriched, showing this table. We found that some pathways are replicate the findings in literature, which were highlighted in this table. For example, the notch thickening pathway, type 2 diabetes. Apart from the linear regression and cake pathway enrichment, we also used the machine learning to find the optimal proteins to discriminate A-beta. As we have measured 4,001 proteins, if we want to identify biomarkers, we don't need to measure that much amount of protein. So that's why we want to find the minimal panel of proteins which can predict the beta pathology. So we used the lasso for feature selection and SVN for classification. We found that only a panel of 44 proteins, along with demographic information, achieved an AUC of 0.178 in group 1, showing the red color in this figure. And the same feature, including proteins and demographic data, achieved an AUC of 0.168 in group 2, showing the green color in this figure, indicating that this 44 proteins has a good performance in predicting a beta pathology. So all these results were published in Alzheimer's Dementia in 2019. If you are interested, you can check more details in this paper. Apart from machine learning, we also used the co-expression network analysis to find the protein modules linked to AD pathology. So the co-expression network analysis means that we classified proteins based on their correlation with each other into different clusters. So we obtained 9 different modules shown in figure A. We then analyzed the association of these modules with AD pathology, including A-beta, p-tau, and neurodegeneration, vascular damage, and inflammation. Because EMIF data has lots of data collection, including all these features, so it gives us an opportunity to identify the protein modules associated with the different AD phenotypes. So as you can see that of the 9 modules, we found M2 blue and M4 yellow modules are significantly associated with AD pathology markers, showing this heat map. In addition, the associations are consistent in the direction of change. For example, these 2 modules are positively associated with A-beta, p-tau, and total tau, but negatively associated with hippocampal volume, which means brain atrophy. So all these results show the consistency between different AD biomarkers. Because these two modules are very important, we then checked the hub proteins, which means the most important proteins within these two modules, shown in figure C and D. Some of the proteins have been reported previously. For example, the apolipoprotein D, FAT3. Some of them have never been reported before. So all these hub proteins within these two modules provide tractable targets for further mechanism study because they are the most important proteins within the two most important modules relating to Alzheimer's disease. So apart from AD pathology markers, we also checked the association of these two modules with AD clinical diagnosis, including control, MCI, AD, showing figure A and B. We also compared the association of these two modules with MCI conversion, which means we compared them between MCI converted to AD and MCI non-converted to AD. It's a disease progression. In addition, in figure I and J, we compared the association of these two modules with MMSE score, which is a clinical diagnosis. So as you can see that these two modules increased in AD compared to healthy controls, increased to MCI converted to AD compared to MCI non-converted to AD. In addition, they were negatively associated with MMSE score. So all these results taken together, showing that there are consistency between the pathological analysis that I showed you in heat map and the clinical diagnosis shown here. So you could check more details in this paper, which was published last year in Alzheimer's dementia, if you are interested in more details. So I just showed you the results from SOMA scan, you having 4001 proteins. And now I'll show you the results we have analyzed based on the immune assay to test 34 previously identified proteins. So we use different platforms to measure the 34 proteins. For 25 proteins, we used the Numinix and ELISA to measure them in 1,000 subjects. This table shows you the proteins, the details. The first column shows you the name of the proteins. The second column shows you the association of these proteins with AD and the phenotypes. All these results are based on our previous results. For the rest of 9 complement proteins, we used the MSD platform to measure them in nearly 900 subjects. These 9 complement proteins were either associated with AD diagnosis or with MCI conversion, shown here. After we get in the results, we firstly analyzed the association of these proteins and a beta group. We used the logistic regression, and we corrected for variables such as age, gender, and ApoE4 genotype. In this table, we found that 7 proteins passed both p-value and multiple correction recorded q-value, which were highlighted in this table. In addition, we found that the proteins replicate previous findings on the change of direction. This means that here we found that phecolin-2, this protein, increased in A beta positive individuals. And in our previous findings, we also found that phecolin-2 increased in A beta positive individuals. The directional change are consistent. After getting the results, the next step we want to do is to build a multi-protein classifier to discriminate a beta group. What we did is that we combined the 7 proteins with age and did the AUC curve. We found that the AUC of this combination is 0.1774, showing this figure. And this AUC is 12% higher than using age alone. Well, this has a high significance in clinical settings because these biomarkers can be used as a pre-selection tool to diagnose patients for the further diagnosis. In conclusion for this part, as I showed you that we used both untargeted soma scan and targeted immunoassay to identify and validate biomarkers in this large cohort. In addition, we used a range of analytical methods, as I showed you. For soma scan data, we used linear regression, machine learning, cluster analysis, pathway analysis, and so on. Third, I want to point out the importance of collaboration, because as you can see, this is a large effort in collaboration with our collaborators. We can't do it. I don't think we can finish it in Oxford in one site because it involves different expertise. For example, involved doctors, clinicians, people who have expertise in doing PET scan, have expertise to doing MRI scan, and metabolomics, and so on. So it's a large effort of the collaboration. So I think if you want to do any biomarker study, I think it's important to include different collaborators who have different expertise. You could do it as the same way as we did in EMIF. Actually, we didn't collect any new data. We only invited our collaborators to take part in the study by increasing the statistical power of doing the cohort and biomarker study. So that's the conclusion for this part. Now, the second study I want to talk about is called GS study, which is a community-based study. GS is a short for Generation Scotland. As you can see, it's a study in collaboration with the Scottish collaborators. So the GS study is a large population-based cohort study, including over 24,000 individuals. So in this study, we selected 1,061 cognitive normal individuals with brain MRI measurement from this large cohort. So this cohort included 628 female, 433 male. We also collected plasma samples from each single individual, and then we did a proteomic analysis using soma scan, which can measure over 5,000 proteins. We then used a different analytical approach to analyze the data. There are two objectives for this study. First, we wanted to identify proteins associated with neurodegeneration and vascular damage in healthy individuals, which means in preclinical stage. We called it early biomarkers because the participants are healthy individuals. The second objective is to explore brain sex-related differences, because as we know that female have a high chance of getting Alzheimer's compared to male. Here we wanted to know whether proteins mediated such differences. So after getting the data, we first analyzed the association of proteins with a hippocampal volume, which is an index of neurodegeneration, and white matter hyperintensities is an index of vascular damage. So in the figure A, we found that these are the number of proteins which are positive and negatively associated with the hippocampal volume, as you can see from figure A. In figure B, we did cake pathway enrichment for this positively and negatively associated with proteins, showing figure B. As you can see that some pathways replicate literature, for example, the axis guidance, cytokine receptor interactions. Previous literature also reported that this pathway is associated with Alzheimer's disease. So these are with the hippocampal volume. In figure C, we analyzed the association of single proteins with white matter hyperintensities as an index of vascular damage. In comparison to the hippocampal volume, we found a few proteins past the significance, which you can see from here. In figure D, we did kick pathway enrichment. Again, we found that 6 pathways were significantly enriched. Some of them have been reported to be in literature. Again, like axon guidance, cytokine receptor interaction. Apart from this analysis, we also did co-expression network analysis, as I introduced previously. So in this data, we've obtained 8 different modules showing figure A. In figure B, the heat map, we analyzed the association of these modules with the neurodegeneration measured by hippocampal volume and vascular damage measured by white matter hyperintensities. Here we analyzed in a different way because we wanted to test whether there's a gender difference. That's why we divided them into female and male, as you can see from here. We found some interesting findings. First, we found 8 modules were associated with hippocampal volume and vascular damage. Second, we found that there's a difference between female and male. For example, the M5 green module was associated with hippocampal volume and white matter hyperintensities only in male, but not in female, as you can see from here. In contrast, the red module was associated with hippocampal volume and white matter hyperintensities only in female, but not in male. So these findings are very interesting. We then wanted to know whether proteins has a causal mediation with such differences. We used the causal mediation analysis, and we found that 4 proteins shown here mediated such differences in hippocampal volume, and 1 protein, apolipoprotein B, mediated such differences in white matter hyperintensities. So these proteins are very interesting targets for further study because there are 6 related neurodegeneration and vascular damage in preclinical stage of Alzheimer's disease. This work was published in Alzheimer's Dementia DADM in last year. If you are interested in, you could check more details there. So from the GS study in conclusion, what I want to talk about here is first that here we used healthy controls, which means we can identify biomarkers potentially could be used for the early diagnosis. And second point is for the community-based studies, you also need to collaborate with the big cohort initiatives. You don't necessarily need to screen all the cohorts, all the individuals in that cohort. You can select a subset of individuals in that cohort and run a proteomic analysis to identify biomarkers associated with disease at a preclinical stage. So that's for the GS study. And now I talk about the meta-analysis. As I've showed you that we have done biomarker study for over a decade. So we have gathered lots of cohort data. So personally, I'm very interested to see whether the biomarker has a robust diagnosis performance. To do this, I did a meta-analysis. So I summarized all the data we have together, as shown in this table. I've summarized 7 studies together. In terms of the sample size, as you can see, it included over 1,000 healthy controls, nearly 900 MCI patients. In addition, we had data for the disease progression, which means we know who is converted to AD patients, who is not converted to AD patients within 5 years. We also have nearly 900 AD patients. And the last column shows you the number of proteins. As you can see that in different studies, we studied different number of proteins. So if we want to do a meta-analysis for one single protein, the selection criteria is that this protein must be measured in at least 4 different studies. So I first checked the meta-analysis of protein biomarkers for AD diagnosis, which means between AD and healthy controls. We found that 6 proteins matched the criteria, showing these 6 figures. Of the 6 biomarkers, we found that 2 proteins, Ficolin-2 and FGG, showed a significant difference between AD and the controls, showing this figure E and F, as you can see from here. While for the other biomarkers, you can see they didn't pass the significance. So this is for the AD diagnosis. We also checked the protein performance for predicting MCI conversion, which means predicted disease progression. We found 5 biomarkers matched the criteria, showing figure A from figure A to figure E. However, we didn't find any biomarkers reach the significance between MCI converted to AD and MCI non-converted to AD, as you can see from here. So this meta-analysis is a very strong method. You can do the meta-analysis based on your own studies, for example, as I showed here. In addition, if you are interested in a certain biomarkers, you can go to literature to search all the performance of of those data. And once you get all this data, you can ask the author to provide you the raw data. I think they will be generous to give it to you, because I have done another meta-analysis. I asked other authors to provide me the data, and I did meta-analysis. So this is a robust tool, which means you can do it on your own data. You can also do it on the public available data. So the last study I want to talk about is a DKK1 study, which combines in vitro and human samples. The study design. Lots of evidence show that Winter pathway plays an important role in AD diagnosis. And DKK1 is the most important proteins in Winter pathway. So our research question is whether a DKK1 induced protein signature generated from in vitro was detectable in human blood. So to do this, we firstly overexpressed the DKK1 in HER293 cells. We then collected the cell license and did a proteomic analysis. Using PLS regression, we obtained top 100 proteins recorded DKK1-induced signature. So that's the signature we obtained from in vitro. We then tested whether this signature were also associated with Alzheimer's disease patients in human plasma. So we measured the association of this DKK1-induced signature with AD diagnosis and cognition. Here we used the publicly available data as well as our own data. We found that one protein has replicated across 2 different cohorts. We then further used the Mendelian randomization to analyze the causal relationship between this protein and AD. So that's the pipeline of the study. And now I'll show you more details in each step. So we first overexpressed the DKK1 by adding DKK1 plasmid in HER293 cells. We then collected the cell license and measured 1001 proteins in the cell license. We then used the PLS regression and obtained top 100 proteins recorded DKK1-induced signature. We further did KIK pathway enrichment for this top 100 proteins showing this table. And you can see that some pathways replicate literature such as Alzheimer's disease, P15-3 signaling pathways, which were associated with Alzheimer's disease. We then checked, that's the in vitro part. Now we checked the association of this signature obtained in vitro with AD diagnosis and cognition in 2 independent cohorts. One is a publicly available cohort called ANM, which included nearly 700 people. Another cohort is EMIF AD cohort that I previously introduced. So in this heat map, we compared the association of these proteins with AD diagnosis here in EMIF cohort and here in ANM cohort. So basically, we compared the AD versus control, AD versus MCI, MCI versus control, MCI converted to AD versus MCI non-converted to AD. So in this heat map, as you can see, of all these proteins, only one protein, C3, replicate between two different cohorts, EMIF and ANM. Only one protein replicate. In addition diagnosis, we also compared the association of these proteins with the cognitive measurement in EMIF and in ANM. So in EMIF, we had more measurements, including executive function, language, memory delayed, memory immediate, MMSE. In ANM cohort, we only have two measurements, including ADS, COG score, and MMSE. Again, of all these proteins, only one protein, C3, replicate in two large independent cohorts. We then were very interested in why, to test whether C3 has a causal relationship with the AD diagnosis. So we used the Mendelian randomization. Mendelian randomization is a very powerful tool to explore the causal relationship between exposure and outcome. So here I give you a brief introduction of the Mendelian randomization. So Mendelian randomization uses instrumental variables such as genetic variants to explore the causal relationship between exposure, here is a C3, and outcome, here is AD. So Mendelian randomization has three assumptions. First, this genetic variance should be associated with exposure. For example, which means that this genetic variance should be associated with C3. And second, this genetic variance should not be associated with confounders, such as ApoE, for example. Third, the assumption is that the association between genetic variance and outcomes are through exposure. Therefore, if we found that there's a significant association between genetic variance and outcome, we can conclude that C3 has a causal relationship with AD. So that's the whole idea of the Mendelian randomization. It is a very powerful tool to explore the causal relationship between exposure and outcome. So based on Mendelian randomization, we did an analysis and we explored the association between C3 and AD risk, between C3 and A beta, and between C3 and a tau. We found that there's a potential causal relationship between C3 and the AD risk, but such causal relationship was not found between C3 and A beta or C3 and a tau, which means that C3 might be causally associated with AD, but not with AD pathology like A beta or tau. So all these results were published in Journal of Alzheimer's Disease in 2020. If you are interested, you could check more details. So in conclusion, first, I'd like to stress again the importance of collaborations as what I showed you here in EMIF AD study and the Generation Scotland study. I think one side is not cannot finish this large cohort study on their own, but we definitely need to collaborate with different people from different institutes. Second important point is data analysis. As I showed you, we used different analytical tools such as machine learning, clustering analysis, meta-analysis, Mendelian randomization. So those are strongly powerful tools to explore the data. Last but not the least, I think it's important to reuse the existing cohort data. As I showed you in the DKK1 study, we did use a public available data, ANM cohort data, to validate our interested pathways and biomarkers. So thank you very much for your time, and I'm looking forward to your questions.